Welcome to the CJD Foundation's virtual conference. As we wrap up this final webinar of our virtual conference series, we thank our hosting partners, the Centers for Disease Control and Prevention, and the National Prion Disease Pathology Surveillance Center, as well as our sponsors, Ionis Pharmaceuticals, Nancy Seipert, Chuck Cash, and Tom Fabian. To view recordings of other earlier webinars, visit our website. Tonight you'll hear from the third panel of scientists who received a 2020 CJD Foundation grant. Our research grants are funded by Family Memorial Grants and by donations to the CJD Foundation and Strides for CJD. If you have questions during tonight's presentations, please type them in during the talks. At the end of the three presentations, our moderator will address questions to the panel of speakers. And now, to introduce tonight's speakers, Professor Richard Knight, who has served at the UK National CJD Research and Surveillance Unit, and as chair of the UK CJD Support Network, and who will moderate tonight's panel.
Okay, so I'm here to welcome you to the USA CJD Foundation virtual conference, the session where the research grant recipients give their reports. This is panel 3. And I'm very sorry personally not to be present in person because I've been to many of these conferences over the years, and I think meeting everyone directly is very much preferable to this, but obviously this is what we have to do at this strange moment in our time. In these panels, people that you, the families and others, have supported through money to do their research have the job of presenting to you what they've been doing in their research. So this is an opportunity for you to hear how your money is supporting research, what researchers are doing, and of course to ask questions. And in this particular panel, there are going to be 3 presentations that all relate to research on possible treatments, which is obviously a very important issue for anybody who deals with these diseases. So with that said, I'm going to move on to introduce the first speaker, Dr. Anna Palegi, who is from the Department of Neurosciences in ISS in Rome, who's going to talk about investigating the influence of a particular gene on the age of onset of CJD as a potential preventative measure. And I just thought I'd make 1 or 2 comments that may help you as families to put this into context. You know that there are different forms of CJD, and in particular there's sporadic CJD and genetic CJD. And the cause of sporadic CJD is unknown, but it's likely that various genetic factors are going to be relevant to this disease, as indeed they are relevant to many, if not all, diseases, even if they don't actually cause the disease. So it's reasonable to look at genetic factors that may be relevant to sporadic CJD. And in genetic CJD, the age at onset of the condition varies, even within the same families who have the same inherited mutation, and it's likely that other genetic factors will be relevant to this. And I think understanding these genetic factors may help earlier diagnosis and better understanding of the disease processes involved, and these are both potentially relevant for treatments. So with that minor introductory comment, I'll hand over to Dr. Anna Palegi.
Thank you for having me today and for giving me the opportunity to talk about my project. I divided my talk into 3 sections. One part showing the background of the study that we published 2 years ago and from which we started the project. And one part showing the specific aims of the project and the results that we obtained despite the COVID-19 emergency, and a part with the future steps. I would like to refresh some important points in the field of prion disease, such as the need to discover new biomarkers, useful for the early diagnosis, for example, for the prognosis, and as a project. First of all, we need to functionally characterize these candidate modifiers. For example, by the study of their expression patterns. Then we need to fine map the locus tagged by the polymorphism identified in the CYP4X1 gene in order to find possible genetic variants in functional elements. And finally, we aim to validate the gene silencing approach by Cas9 and CRISPR technology and its potential use in animal models for further studies. We started analyzing the expression pattern of these modifiers in the brain of sporadic CJD and control samples. As we can see in this slide, CYP4X1 gene is the only gene less expressed in the brain of sporadic CJD than in controls. And this is important because it is conceivable that the polymorphism identified in the genome-wide study exert his effect, his influence on the age of onset of the disease by modulating the expression of the CYP4X1 gene. Then we checked if this gene is also differentially expressed in Creutzfeldt-Jakob and control subjects, also in blood, that is a more accessible tissue, but we found no differences. Then we wonder if the differential expression that we observed in the different tissue of blood and brain could be explained by a differential tissue-specific expression of isoforms of the CYP4X1 gene. The gene has 3 isoforms that are RNAs of different length, and in the experiment we found no differences in the expression of these 3 isoforms in blood and in brain, but one, the isoforms number 3, that is slightly less expressed in the brain of sporadic CJD than in control samples. But this data need further validation because the expression of the CYP4X1 gene in the brain is very, very low. And so we need further experiments. Then we wonder if the regulation of the expression of the CYP4X1 gene could be explained by the presence of allelic imbalance phenomenon. That is when the allele of one gene is expressed, so it forms RNAs and then proteins more or less than the other allele. And we investigated this hypothesis by according to published protocols. So by the sequencing, the Sanger sequencing of RNA. We considered only heterozygous brains of at the polymorphism of interest that unfortunately was located in an intron region, that is an area of the gene that is not transcribed, so doesn't form RNA or protein and then proteins. So we had to choose other 2 polymorphism in high linkage with the the polymorphism of interest. That means that I linkage that they behave in a similar way. So if I have a variation, a nucleotide variation in my polymorphism, I have a similar variation also in the other two polymorphism. This polymorphism is located in an exon region, so they are transcribed. And if the allelic imbalance phenomenon is in place, is really in place, I should observe at the DNA level 2 peaks at the sequencing corresponding to the heterozygosity of the polymorphism. And at the RNA level, I should observe only 1 peak, so the prevalence of a peak corresponding to the most expressed allele RNA. And this is exactly what we observed in 8 brains heterozygous for the CYP4X1 polymorphism, and in the 2 polymorphism in the exonic region and in linkage. We observe at DNA level, obviously, the heterozygosity, while at the RNA level, we observe the preferential expression of the normal allele. And this is an indirect indication that the risk allele A of the polymorphism in the CYP4X1 gene is downregulated in our samples. So it is a low producer allele. This data was also reinforced by a preliminary immunohistochemistry analysis performed on brains of sporadic CJD with the homozygous genotype for the normal allele and homozygous genotype for the risk allele. And as we can see by these figures, the CYP4X1 protein is less expressed in the AA genotype. This analysis needs to be confirmed in next experiments. Then we went on performing the whole transcriptome analysis on the brain of mutated CJD. And in this slide, I represent the different steps needed for the experiment, starting from the sample selection, the RNA extraction, the steps to prepare the sample for the sequencing, the analysis of the raw data with the quality check, and finally the differential gene expression analysis. And we compared patients with extreme phenotypes, so 4 late onset of the disease versus 4 early onset of the disease. And as we can see by the volcano plot and the MA plot, we obtained 6 deregulated and statistically significant transcripts. Most of them are pseudogenes, and there is also a zinc finger protein that are well-known regulator factors of the expression. And this experiment needs to be completed, and we will start next month the second run of sequencing. And in fact, in conclusion, next week, at the beginning of November, we will run a second experiment of RNA sequencing with additional 8 brains of mutated CJD. We will similarly start with the genome sequencing, the whole genome sequencing. We have selected during this month around 20 samples, mutated E200 key mutated cases, and we think to finish these experiments in 2-3 months. And finally, when we have completed the AIM2, AIM1 and AIM2, we will start with the experiment, the sciencing experiment in cell samples, in cell cultures, and I think we will finish this part in around 4 months. So thank you so much for your attention. I would like to thank all the contributors to the funding of my project, the Robert Dodd, the Patrice Agorty, the Mark Gardner Memorial Grants, the Rights for CJD Grant, the International CJD Awareness Day Research Grant, and of course I would like to thank also all the collaborators to the project at the Istituto Superiore di Sanità in Italy, the University of Bologna, Bambino Gesù Ospedale Pediatrico, and the University of Verona. Thank you so much.
Okay, now it's my pleasure to introduce Professor Qingzhong Kong, who is Associate Professor in the Department of Pathology at Case Western Reserve University. And he is also, of course, the Associate Director of the National Prion Disease Pathology Surveillance Centre. And he's going to talk on a subject he calls the RAP approach for effective prevention and treatment of prion diseases. And again, I thought I ought to make 1 or 2 preliminary comments just to try and frame this for you. The basic molecular underpinning of prion disease, as I'm sure you've all picked up by now, is that a normal protein called PRPC converts somehow to an abnormal form of protein, PRPSC. That's the molecular underpinning. But as an illness affecting people, people become ill because neurons, brain cells, become sick and die. And it's very important to register the fact that the cause of the neuronal damage, the relationship between this molecular change and what happens to neurons to make them sick and die, is not fully understood. On the basis of all of this, people have suggested 3 general and potentially distinct treatment approaches. First of all, reduce or get rid of PRPC, the normal prion protein, because if you get rid of it, there's none there to change into the abnormal form. Or you reduce the abnormal form, the PRPSC that's being made from PRPC, or you somehow reduce neuronal damage if you understood or understand what's causing that neuronal damage. And people have tended in their treatment approaches to center on one of these, but I think the interesting aspect of what you're about to hear is the idea that perhaps you should try to concentrate on all 3 approaches. So with that, I'll hand over to Professor Kong to explain his work. And again, just to remind you, if you have any questions, to type them into the box provided.
Good morning, everyone. I first want to thank Dr. Richard Knight for giving me a kind introduction, and once again, CJD Foundation for giving me opportunity to give a report of our effort trying to develop a comprehensive approach for effective prevention and treatment of human prion diseases. Prion disease, as many of you know, is a group of fatal transmissible disease that requires the cellular prion protein, or PRPC. Unfortunately, there's no treatment available at this point. Purification not only requires PRPC, but also requires PRPC as the mediator or receptor for its toxicity. And more importantly, or equally importantly, PRPC appear to be dispensable because in knockout mice or other animals knockout with PRP knockout, there appear to be no big problems. So this makes PRPC the ideal target for therapeutic intervention. Cellular PRP is a small protein. As shown here, an N-terminal part is a flexible region of her flexible tail. C-terminal part is structured globular domain, and normally goes through different kind of processing or cleavages. I want to highlight the alpha cleavage, which cut almost the middle of the protein, which will destroy the capacity of the PRPC as a substrate for purification. In the meantime, produce two beneficial fragments, N-terminal N1 and C-terminal C1. N1 has been shown to be neuroprotective. I'll talk about more later about it. And the C1 has been shown to be able to inhibit prion. Prion disease is complicated and diverse. In humans, the primary form is the CJD, which can be subclassified into sporadic form and familial and acquired forms. The sporadic form also has multiple subtypes. So this, because there are other types of human prion disease. This huge complexity and diversity among human prion disease patients is a huge challenge when you try to develop a therapy against all human prion disease. So the efforts to develop a therapy against prions can target different aspects of the prionification, clearance, or pathogenesis. Basically, it can try to reduce the cellular PRP level, which is the substrate, the PRP substrate, and also receptor for toxicity. And second, it can try to inhibit the prion agent PRPSC replication. It can also try to enhance the clearance of the PRPSC, or it can prevent toxicity presented by the toxic molecules during prion pathogenesis. And it also can try to restore or regenerate the loss of neuron or neuronal functions. And finally, it can try to inhibit neuroinflammation that has been shown to be important in prion disease pathogenesis. So how to achieve those goals? You can do either by small molecules, which is commonly used by most pharmaceutical companies, try to affect any of these processes. Secondly, because anti-PRP antibodies, which will try to knock down PRP level or clear PRPSC. And you can also use the siRNA, which will try to knock down mRNA encoding the PRP gene to reduce the PRP levels. The same goal can be achieved by using antisense oligonucleotides, which has been shown great potential in animal models studied recently. And you can also use stem cell therapy to replenish the lost neurons or other brain cells, try to restore function. And you can also use gene therapy to achieve any of those goals here. And finally, you can try to develop a vaccine that will prevent or eliminate the prions before or during its replication. However, there are quite a few drawbacks of the current efforts up to this point. First, most studies have been done using non-human prions, basically use animal prions, and human prions may be different. And second, they usually only target one or two aspects of the prion pathogenesis. Unfortunately, as I told you before, none is proven effective in clinical trials so far. And even if it works, most of the strategy will also require frequent and long-term dosing. So I'm trying to develop a comprehensive approach that will reduce the PRPC level, attenuate the PRP assay level, and protect against neurotoxicity at the same time. And then we're going to use human prions, basically CJD prions, and study them in human neuronal cell line or humanized mice expressing human prion protein, so that the result can be more easily translatable to clinic if it works. So there are three elements to our approach. We call it RAP. First is use doxycycline, which are given orally. Doxycycline is an antibiotic that has been shown to be effective in prion animal model studies. And in clinical trials, it's safe, but did not show protective effect, unfortunately. And the second element is the using anti-PRP siRNA constructs to knock down PRP level in the brain or in the central nervous system in general. Let me explain what RAV is. RAV is a recombinant adeno-associated virus, which is the commonly used viral vector for gene therapy experiments and for some clinical approved gene therapy treatments. It is good because it's safe and it can achieve long-term expression after one shot. And it can be also made into plasmids that can be used as a plasmid therapy if you have some other reagent to help plasmid DNA get into the cell effectively. That's why we choose recombinant AAV vector for our experiments. And the third is using the expression of a secreted form of the neuroprotective PRP N-terminal fragment, which will also be mediated or delivered just like the PRP and shRNA using a recombinant AAV adeno-associated virus vector, which has been shown to be the safest viral vector so far for gene therapy. And the last element, which is not directly associated with the study, but we are trying to develop novel nanoparticles, which will enable safe and efficient in vivo DNA delivery to achieve gene therapy, to bypass the need for viral vectors, which have been shown to be very expensive and carry other safety risks. So in terms of reducing the PRBC level, we'll use several anti-human PRP siRNA constructs that we carried on the recombinant AAV vector. And recombinant AAV siRNA vector will be either made into viral particles, which is the traditional way to do it, or we can directly use the naked plasmid DNA and carry this siRNA and use them in conjunction with our novel nanoparticle reagents to deliver them efficiently into the brain or spinal cord. Then these RAV vectors will be introduced in the brain or spinal cord, the space where the cerebral spinal fluid can flow through the entire central nervous system through the cerebral ventricular or intrathecal infusion. This way is to bypass the blood-brain barrier so that they can reach the nervous system tissue to achieve its effect. To attenuate reduce the PRP assay levels, we achieve it through three ways. One, as I said before, is use oral administration of doxycycline, which has been shown to directly inhibit prion replication. Second, by knocking down the PRBC level using shRNA, we'll also knock down the substrate, which is the PRBC, for prion replication. And the third, by expressing the secreted form, the N-terminal fragment of PRP, we'll show later that actually this N-terminal fragment can also inhibit prion replication. So these three ways will all come together to achieve the goal of reducing PRBC levels, PRP assay levels. So where we are on this, we have started oral feeding of doxycycline-laced rodent food to either specifically MM1 subtype or M1 subtype inoculated human and mice. And second, we have generated the anti-human PRP SH plasmid constructs, and they are being examined for the efficiency and specificity for knocking down human PRP expression in human cell lines and in human and mice. And we have shown, we'll show a little bit later, that the N-terminal fragment of PRP can directly inhibit at least the prion-seeded PRP amplification in archaic assay. So in terms of protection against neurotoxicity, it will be done in two ways. One, we achieve that through recombinant AAV-mediated high-level central nervous system expression of the secreted form of the PRP N-terminal fragment. And second, it can also achieve through siRNA-mediated knockdown of the PRP gene expression, because this will reduce the PRBC level. The PRBC not only is the substrate, but also is a mediator for prion toxicity. So why do we choose the PRP N-terminal fragment and use that to achieve neuroprotective effect? N1, as I showed before, is the N-terminal fragment normally produced through alpha cleavage of cellular PRP. It's shown a good, strong effect to prevent cell death in culture cells. And it's been shown to also bind to toxic A-beta oligomers. That is the central player in Alzheimer's disease pathogenesis. And by doing through binding, they can neutralize toxicity in Alzheimer's disease. And it may also do the same thing with the other toxic molecules, like in Parkinson's disease, maybe in tauopathy even. And the work from our collaborator, Dr. Wenchen Zhou's lab, have shown before, recombinant PRP seem to be able to inhibit preamplification in PMC reactions. And this effect seems to be largely reside on the anti-mineral fragment of PRP. So we hypothesize that the PRP anti-mineral fragment will also protect against the prion toxicity. And I'll show you later that we've shown that the A1 can also inhibit preamplification directly in RD quick regression. So here's our article data showing that if you add recombinant A1 to the prion seeds before you start the RD quick reaction, it can achieve, it can prolong the lag phase of the seed amplification. When you have less seeds, the effect is more dominant, it is more prominent. When you have more seeds, when your seeds concentration higher, the effect is still there, but it's not as dramatic when you have less prions. So before I had this grant, we actually tried a quick third experiment before, where we just introduced the recombinant AAV carry the A1 gene intramuscularly to wild-type mice, and then inoculate the mice with mouse scrapie, and see what happens. Unfortunately, I see that the recombinant AAV A1 injected mice, they showed a slightly longer incubating time, but it's not significant. But one good thing is, at least did not make things worse. It seemed to have some slight effect, but it's very uncertain there. But then we look at whether we can detect the A1 expression from that RAV A1 factor. And unfortunately, we cannot see it when you introduce that gene into the C2C12 muscle cell line. We cannot detect A1 in the medium. And this is not surprising, since after that we learned that from Alex Mippen and his colleagues, result that the direct prion A1 probably is not going to work because it's going to stuck inside the cell, cannot get outside. So to address that problem, we design a larger A1, we call A1 plus, which we show that it can be detected if we introduce the muscle cell line, can detect it in a large amount in the culture medium. We repeated that later again in another experiment, in many subsequent experiments. More importantly, we recently introduced this recombinant AAV A1 plus construct into mice by intraventricular infusion. And we then collected plasma. Here shows that at 4 weeks after the infusion, we can detect the A1 plus fragment in the plasma of the injected mice. And it's not, this same thing is not showing up in the mice that injected with control RV GLP plasmids, showing that it's a specific, only when you inject the A1 plus construct. So we encouraged by this result, we started 2 animal experiments. One, we're using the TGA6H mice, which express human PRP with no glycans. And we show this because it has a very short mean time with the MM2 substrand of human sport CJD. That's just a little over 2-month period. We inject these mice first with this RAV particles, RAV plasmid, with either A1 plus or the GLP control. And 3 weeks later, we inoculate them with this pre-adapted MM2 prion strain. And now I'm waiting for the symptom to occur, and then to check for their pathology and the PRP accumulation. The second experiment we did is using the T40H mice, which expressing wild-type human PRP. And we showed if we inoculate this mice with the common MM1 substrand of CJD, it has about 6 months of incubating period. Now we're injecting animal in 3 groups, two of them with the N1 plus construct, the other with the GIP control construct. And after that, 3 weeks later, we inoculate with M1 prion. And then one group will be given doxycycline 1 week later. And now we are also waiting to see whether any of the treatment have protective effect on the prion disease progression. And this is where we are at this point. We are planning to further test our shRNA construct to show their specificity and effectiveness in neuronal cell lines and human mice. And we're going to, once that's done, we're going to use the best shRNA construct to treat them, to use them to treat our mice, human mice, either alone or in combination with the A1 plus construct and oral doxycycline. And we're also going to try to further optimize and validate our nanoparticles so that we'll be able to achieve affordable and safe, effective in vivo gene delivery to patients affected by either prion or potentially other disease like Alzheimer's disease. Finally, I want to thank the people in my lab, especially Qingzhong, Manuel Camacho, and several very talented students, and also Qi Xu for conducting this work. And my collaborator, Dr. Wenchen Zhou, and Dr. Wilder Schultz, who provided the recombinant N1, and for the Preventive Pathology Surveillance Center, which helped with our quick experiments. And I appreciate all the families affected by CJD, and also I want to thank definitely the funding provided by the CJD Foundation and the Strides for CJD, and the families who generously provide the funds, the Gary Butterman Memorial Grant, the Michael Cole Memorial Grant, the Ernie Donahoe Memorial Grant, and the Priest Family Memorial Grant, Research Grant, the Tom Stevenson Memorial Grant, and the Teresa A. Strauss Coolidge Memorial Grant. This funding has made it possible for me to jumpstart my long-term dream trying to develop therapies to treat human prion disease, because I see the suffering of families, and I actually been contacted by a few families who I never know of, tried to ask me for help, but I felt bad I could not help them. But hopefully this funding with our starting project, I hope in the near future we'll be able to help them. And thank you for your time and attention.
So now I'm going to introduce Dr. Natalia de Carmo Ferreira, a postdoctoral fellow from the Rocky Mountain Labs, who is going to talk about the evaluation of new anti-prion drugs, from drug design to therapeutic efficacy. And I'll just make a few framing remarks again, as before. Of course, people want treatment for prion diseases, and people may suggest certain treatments or even develop certain treatments, but the real question is, do they work? And obviously a subsidiary question, which is maybe not so obvious, is how you determine if they work. And if you have a chemical compound, you could study it chemically just by its actions at a chemical level, but that's not very biological, and you'd like to test it in something that actually is living. So you might grow cell cultures, cultures of cells in a laboratory, and use them. But of course, cells in organisms don't live on their own. They live in organs like the liver or the brain. And so people have developed sort of miniature organs, what they call organoids, in laboratories. But beyond that, of course, organs live in whole biological creatures. And so people tend to move on to testing in animals, and then, of course, on to the real test of testing in humans. But quite often, before testing in humans takes place, some of these other kind of testing procedures are used. So that's really the subject that's going to be addressed here, how to evaluate possible new anti-prion drugs via these various methods. So I'll hand over now to Dr. Natalia de Carmo Ferreira. But of course, as before, if you have any particular questions, please type them into the box.
So first of all, I would like to thank to all of you who donated and who strided for this grant, and to say that it means a lot for me to be awarded with this grant and to contribute with my research. So my project consists in the evaluation of new anti-prion drugs which were planned and synthesized for the purpose of this study, and to combine different approaches to find new drugs to treat prion disease. So, as you all have heard, the main event in prion disease is the conformational change of the prion protein, the normal prion protein, which we call PRPC or PRPSAM, to the pathogenic isoform, which we call PRPSC or PRPRAS. So the cells in our body produce the normal prion protein, but once the PRPSC is present, this pathogenic isoform is then able to convert the normal prion protein and make new copies of the pathogenic isoform, leading to the accumulation of PRPSC both inside and outside the cells. And the ones that are outside are then available to infect other cells. And this is how the disease is spread through the brain. A really important characteristic of prion disease is the existence of different prion strains, which means that the PRPRAS can adopt different conformations, which means different shapes. So, and each shape is associated with a prion strain. So in the lab, this means that we can find different results because the different strains have different behaviors. And biologically speaking, it means that these different strains will tend to accumulate in different brain areas, and this will lead to different clinical signs. So this is why it's so difficult to find a drug that works for different prion strains. So there are many reports in literature from different researchers showing that we can find a drug that works for one prion strain, but unfortunately it doesn't work for another. So this is why it's important to assess different prion strains. The drug discovery process for prion disease comprises different stages. So overall, more than thousands of compounds have been tested by many researchers in the world. And then, based on the initial results, the researchers proposed some chemical modifications in the structure of the drug, aiming to find drugs with better characteristics, such as more with solubility. So then hundreds of them have been tested in vivo in prion-infected mice, but here is where they fail, and for many different reasons. And the main ones are because they are toxic or because they don't reach the brain, which is the target of this disease. And then to date, less than 5 drugs got to the point to be tested in humans infected with CJD. And unfortunately, none of them really worked. And this is why these diseases are still untreatable. So this highlights the difficulty to translate the results we get in vitro and to in vivo to animals, and then from in vivo to humans. So, and another important thing here is if we are able to use human prion strains in early stages of research, this can increase our chances to find a drug that will work in the end in humans. So where did my project start? So a few years ago, I analyzed a library of 200 compounds, and by using different approaches, I found this lead compound that we call J8. And based on the structure of these compounds, my Brazilian collaborators planned and synthesized a new series of compounds that we call LCs. So the goal of my project is to combine different approaches such as prion-infected cells, cell-free assays, where I can test human prion strains, and the newly developed model of human prion infection, which are the cerebral organoids, and lastly, to test the prion-infected mice in order to find an anti-prion drug that will work in vivo, in humans, sorry. So the first model I used are the prion-infected cells. So these cells are derived from a mice tumor, and this is why they are able to grow indefinitely in a dish, and they are infected with prions. So this is what happens in the absence of treatment. We have a really strong signal of PRP-RAS, but in the presence of the different drugs, we see an inhibition of the accumulation of PRP-RAS, which is showing here in this graph. So here are the untreated cells, which have a lot of PRP-RAS, but in the presence of these drugs, they have few amounts of PRP-RAS. So by using cells infected with the RML strains, these drugs were able to reduce by more than 70% the amount of PRP-RAS. However, when I tested the same cell but infected with another prion strain, which is the 2N2L strain, only few compounds were effective, and even though their effect was weaker than in the RML strain, reinforcing that some compounds can work for a given strain, but they are ineffective against others. So then I switched to another model, which is a cell-free assay, where I could test a human prion strain. So in a tube, I added a brain homogenate from a patient with CJD, and I added the normal prion protein because, as I told you before, the infectious prions in the CJD brain homogenate are able to convert the normal prion protein, and this is what is showing here. I have a really strong signal of PRP-RAS, and here are the same reactions, but in the presence of the different drugs. And here, LC19 and mainly LC87 were able to reduce or inhibit the PRP-RAS formation in vitro. And again, here I used a human CJD prion strain. But we have to keep in mind that this is a cell-free assay. And in a cellular environment, many other factors can interfere with the interaction of the PRP-SC and PRP-C. So this is why it's so important to have a human model where we can test the efficacy of these drugs in earlier stages of the research. So here, I used the cerebral organoids. So I've been doing these experiments in collaboration with Dr. Catherine Hai and Bradley Grooveman, who developed this new system. So these cerebral organoids, which are also called mini brains, are derived from skin cells collected from patients. So by adding different factors in vitro, we are able to induce the formation of this three-dimensional tissue, which have some characteristics of the human brain, and this is why we call them mini brains. So then, by adding a brain homogenate infected containing CJD prions, they showed that in the first month, they had low or no detectable levels of PRP-RAS, showing that these mini brains get rid of the inoculum. There was no residual inoculum there. But weeks later, they found new generated CJD, PRP-RAS. So this is the most recent model of human prion infection. So then I decided to assess two different regimens. The prophylactic treatment, where we can treat these mini brains prior to the infection, which mimics what could happen in the context of genetic prion disease, where we can treat these patients before the appearance of the symptoms. So here, I started the treatment. I exposed these mini brains a week before infection to two different drugs, the J8, which is my lead compound, and PPS, the pentosan polysulfate, which is a non-anti-prion drug, which works in cells, in animals, but unfortunately didn't work well in humans. So, and then I removed the treatment. I exposure a week before the infection, and I kept for 2 more weeks, and then I stopped the treatment. And then a month later, I measured the amount of PRP-RAS in these mini brains. So in the first month, I couldn't see any big difference in the treated and the untreated mini brains. But 2 months later, I saw that the mini brains, which were treated with PPS, they had 10-fold less PRP-RAS compared to the untreated ones. The J8 inhibit a little bit, but it's not significant, statistically significant. So then I assessed the late treatment, which is the therapeutic treatment, which is similar to what happens with CJD patients, where we treat these patients, but the disease is well established. So they have the clinical signs of the disease. So here I start the treatment 2 months after these mini brains were infected. And then I treated these mini brains for a month, and I measured the amount of PRP-RAS. And as we can see here, both PPS and J8 were able to reduce the amount of PRP-RAS in these mini brains by more than 100-fold. And then 120 days post-infection, which means 2 months later, even though these mini brains were not being exposed to the treatment, the ones that were treated with PPS still had more than 100-fold less PRP-RAS than the untreated ones. And the ones which received the J8 also had less prions, but again, this was not statistically significant. But we have to remind that this was just a brief treatment. Now we are performing this in a long treatment all the way to the end. So this highlights the utility of this new model of human prion disease to access the efficacy of anti-prion drugs. So to summarize my data, we showed that these three-dimensional cell culture, which we call mini brains, are useful models to verify the ability of anti-prion drugs in inhibit the conversion in a human background. And we hope that by using this human model, we can increase our chances to find anti-prion drugs that will work in humans. So our future goals are to test the LC's compounds in this system in the mini brains infected with CJD, and lastly, to test the effective drugs in humanized in transgenic mice that are able to be infected and propagate prion infection when they are inoculated with CJD prions, which are human prions. So I would like to thank you again to the CJD Foundation, to the families who contributed for this grant. So the Dodom in the Dorf, the Jeffrey Smith family, as well as the people who strided for this grant. Thank you all. As well as my Brazilian collaborators who designed and synthesized these drugs. So Irama Cordeiro, Maria Letícia Barbosa, Arthur Camerli, as well as Bradley Groveman and Catherine High, who taught me how to grow and generate these mini brains in the lab. And of course, Dr. Colhay, Byron Colhay group, where I work at NIAID. Thank you all.
Okay, thank you very much for the three presentations. The final message that came through on the question front said, "This is really interesting. Thank you very much for your research." So that's at least a nice bit of feedback for you all. I've got various questions that have come through. So I thought I might quickly go through the specific questions that have come through for each person. So for Dr. Paleggi, perhaps a very difficult question. What is the most likely explanation that a 94-year-old with an E200K mutation has never become ill?
This is a very good good question, and we are asking this for at least 4 or 5 years in our work. And yes, this is fundamentally the question of my research. We observe
they're asking you what do you think is the most likely explanation? I know you don't know. Yeah.
Yes, the most likely explanation is that there are something else. So some environmental or genetic factors that are in place together with the PRNP protein, and so the genetic risk of having a mutation in the prion protein gene. So we observe in our family, in Italian, but also in other clusters such as the Slovakia cluster, that there is a not complete penetrance in the E200K mutation, and this penetrance is maybe is explained by other factors. So the discovery of these modifiers of the penetrance rate of the age at onset is essential for understanding the underpinning mechanism of the pathogenesis of the disease, of the progression of the disease, and also they could be very important for the early diagnosis, for the also for the hypothesis of a new therapeutic approach that is preventive and not curative of the disease. So I think I really believe that there are some other genetic factors that are in action.
I think what you've said in effect is that there are likely to be other genetic factors which that influence the E200K mutation. Yeah. And you hinted about environmental factors, which I guess is just as an interesting point for me. The question always has been as to whether genetic mutations cause the disease, full stop, or whether they are in some way risk factors for particular risk factors for some common environmental phenomenon. And since all disease is an interaction between the environment and the gene in the end, just briefly, do you have any speculation about what these environmental factors might be? Because no one's ever really identified any.
Yeah,
you can say
no. Yes, the finding of environmental factors planning together with genetic factors is a really difficult issue to explain because environmental factors are an enormous, large variables, and we need a very strict way to detect them. We tried to lower the environmental background, studying this cluster, these isolate families that belong all to a very restricted geographic areas that are isolated, and but probably there are also environmental factors that are in place, and I think, for example, all the epigenetic phenomenon that implies the effect on environment on the gene, so on the expression of the genes. So we need also to perform epigenetic studies to understand if there are also this phenomenon in place that it is plausible and conceivable.
Okay, I guess someone also asked a question, is the CYP4X1 gene expressed similarly in everybody who has the gene? I mean, I'm not quite sure what they meant by the question, but I mean, I think the point you were making was that this is a normal gene, isn't it? That everybody has, but exists in different forms.
Yes, yes. Yes, it is a normal gene. And I think that the meaning of this answer might be if the gene is related with the aging, and so if in normal population, in general population, there is fluctuation in its expression correlated with the age. But we found this from the genetic point of view. We found that the genotypic distribution of the polymorphism is the same in the general population and in our population. And the expression in the brain of controls that are neurodegenerative subjects and neurological subjects, brains. So the expression is similar in all the individuals. We have not checked obviously the expression in the brain of normal population, but in GTEx we found that there are not differences in the expression of this gene in the brain in the general population. So it is specific. It seems to be specific for the CJD.
I suppose one of the difficulties I have about the genetic influences on disease, which I don't know what you think, but there are certain genetic things like mutations or the codon 129 polymorphism, which have a very big effect. But there may be many genes that have very small effects. And it may be that there's a certain combination of many genes with small effects that may make that puzzle very difficult to solve, mightn't it?
Yeah, and this is also the reason why it is so difficult to find genetic genes with a little effect in genome-wide studies, because we to detect this factor, these genetic factors, we need an enormous number of samples, and this is a difficult issue in a rare disease such as Creutzfeldt-Jakob disease. So we need to choose the population in study that is really homogeneous. So our approach is that. And in this way, we hope to lower all the variability that we can have in a sample set, and so to empower our studies, our research. Yes, there are I agree with you that there are, I think there are many genes with a little effect that are active together with the main gene that is the prion protein gene.
Okay, well, before we move on to the next topic, there are two things that come through which I think I can answer, although if you disagree with me, then join in. The first is, can you have a pathogenic mutation of the prion gene and not die from CJD? And the answer is yes, that's exactly what you were talking about. There are people who have the mutation and die of other things. Their penetrance is not 100%. And do you find geographical locations that have higher rates of CJD than normal? And I think the answer to that is, well, there are geographical clusters, as indeed you've mentioned in your presentation, which are basically based on familial clustering, because families live in the same area and therefore genetic diseases co-localize. And other than that, as far as I'm aware, there's been very little that's come out of major epidemiological research to suggest that there are geographical locations of any note that require explanation by other means. The distribution tends to be fairly random, as far as I'm aware. So if we go on to Professor Kong, yes?
Sure.
Sorry? Oh, yes.
Sure,
Okay.
I said, sorry.
Okay. So the questions that have come through in relation to your presentation, somebody asked, what does in vivo mean? So these terms ex vitro, in vivo, and in silico, and various things were used. Do you feel up to giving a brief explanation of what they all mean?
Yeah, in my mind, the in vivo general, I mean, in our case, animal experiments, doing experiments in animals.
Yeah.
In vitro means basically either in test tubes or doing in culture dishes. I guess ex vivo probably just some tissue slices like Natalia just mentioned in her study, I guess.
Okay. And there's a question here which you can all join into if you want, but I suspect that none of us are able to give an answer. And that is, how does the abnormal prion protein cause the normal protein to change shape and convert? This of course could start a very long discussion, but I presume the basic answer is we know some things, but we don't understand it completely. You all in agreement with that? We don't really fully understand the process, yeah?
Yes, you're right. I agree with that.
Okay, so the next question that came up, which was really addressed to you again, because you mentioned about doxycycline's efficacy. So someone said, well, if indeed it is efficacious, why aren't doctors giving it straight away when the diagnosis has been made? Well, I guess my answer to that is that I do not know of any evidence that doxycycline is an effective treatment on its own in human beings. It may or may not be a useful preventative treatment, and there is a long-term study going on, Italian study, isn't there, of treating people who are perhaps mutation carriers to see if doxycycline can perhaps delay or prevent the onset of disease. So I think that maybe, do you want to address what you meant in your presentation by saying that doxycycline had efficacy?
Well, I meant that doxycycline had efficacy in animal studies. And in humans, as you mentioned, in human clinical trials, used alone, it may have no effect. Or maybe in one of the trials seem to show some a small protective effect if you give it early, in patients who are early in the stage. So maybe what I'm saying in our case, if we use it as one of the elements, not just one, not only, then it may actually help further improve the efficacy if we use in combination with other approaches, like our gene separate approach.
I suppose some people, and maybe that's the drive behind the question, is that, well, if it might work, why not just give it anyway? And I guess my answer to that would be the history of medicine is full of things that might work and look as though they might work, but don't work, and actually sometimes make things worse. They actually do harm people. So I don't know whether would you recommend giving doxycycline to a newly diagnosed patient?
Well, that's the question. I'm not a physician, but as I said, a few patients did contact me about ask for advice. I told them that maybe doxycycline at least is as safe in animals. In humans, it's safe too. It may not be effective, but does not do much too bad things. So maybe if as a comfort therapy, maybe you want to try it. Probably.
I think it's an individual decision, isn't it, between a physician and a patient if a drug is not
true. Yes.
Work. But I think the point was that you were talking about a specific effect in a certain animal experiment, I think, weren't you? Yeah.
Yes. But what I'm saying, we need to use together. But none of the therapy people try so far in human trial worked. Yeah. So my guess is if just want anything alone may not work. No. In humans.
Well, that was one of your main messages, isn't it, really?
Yes, that's exactly what I'm trying to get through.
The approach itself will not be effective. So another question relates to knocking out PRPC. And you made the statement, I think very carefully, that it seems on the whole not to be very harmful to mice. I just wonder what you think, though, that knocking out PRPC in mice, in nice laboratory circumstances, being looked after very nicely in animal houses, might not really reflect what will happen if you knock out PRPC in human beings in the real world. What do you think about that?
That is true. In mice, the effect of knocking out PRP is limited. Mostly people reported the effect in the long-term potentiation has some defect. But overall, the animal is not too bad. And also there are knockout cattle. To my knowledge, they don't seem to show any big problem either. So my guess is if the person already is at risk of already have prion disease, maybe the risk of knocking out PRP is not that big compared with the consequence of without treating any treatment. So probably it's worth try as for patients who already suffer from CJD.
Yeah, I agree with that, but I just suppose that the point that I feel is that people quite glibly say that knocking out PRPC won't have any bad effects, and I think that one has to be a little bit cautious about what the effects might be in a human in the real world, knocking it out for a long period of time. But
I totally agree with you, because the PRP normally has quite a bit of protective function and other cellular functions. So generally, if you knock out PRP, maybe there's some other genes kind of will compensate for the loss of PRP, but in long term, it may have some problem.
Yeah, and of course, the thrust of your talk was that you wouldn't rely on one particular method alone. So that brings me to the final sort of question, really, and that is that you talked obviously about the third element of this, protecting neurons. And I suppose my question would be, do we know enough about neurotoxicity, why neurons become ill and die, to make that third element of your platform very solid?
There have been quite a few studies, not on prion, but on Alzheimer's disease, showing that a PRP can protect, anyone can protect against that toxicity of A-beta oligomers. And we know that the PRP can also bind to alpha-synuclein oligomers, maybe tau oligomers, and the binding site problem also at N-terminal of the PRP. So there's a good possibility, not solid, totally error-proof evidence that it may work, but it has a good chance it may work. And we know that if you neutralize the toxic oligomers by binding with the free floating A1 fragment or even free floating PRP molecule, if it's not attached to cells, then it will prevent them from going to bind the receptors on cells. That's, I think, they may prevent toxicity from those toxic molecules that floating outside in the cell, in the living cells.
Okay, so we've got 1 minute before I move on. Perhaps the worst question of all, and that is, okay, so you might have somebody who's diagnosed with an illness like CJD, and you're going to treat them, but by the time they're diagnosed, they are a long way down the disease process, in all honesty. So you kind of mentioned, well, we might halt or slow down the disease process and then repair things with stem cells. I mean, how realistic is stem cell therapy in the near future in the context of a panencephalopathic disease like CJD?
That's a very tough question. For one thing, we have two things we want to do. First, you want to prevent toxicity of existing that will cause more damage by the existing toxic molecules. That's the N1 will probably help with that. But if you want to restore the already damaged neuronal function or already lost neurons, you have to rely on stem cell administration to give them new cells to start with. And to my knowledge, that the neural stem cells have made quite a bit of advance in the last couple of years, especially for mining produced cells like oligodendrocytes. There have some advancements that can show that it actually can replenish those type of neurons, neuronal cells in mice at least. So there is some hope, but the question is, even if the neural stem cell can survive after you give them to the patient's brain, whether again to make the right neuronal connection to restore function, that's a different story. So that's still need to be seen.
I think it's a bit further away than people.
Yeah, but I'm hopeful. Probably in the next 10, 20 years, we may have something in the clinic, hopefully.
Okay, so we'll turn to Dr. Ferreira. So someone asked, how did you evaluate the first 200 compounds?
So I used pre-infected cells, and then I moved to some RT-quick assays with CJD strain. And then I also used in silico approaches. So we use different software to predict how these drugs will work in vivo. So they compare the structure of the drug that we are testing with known drugs that were already tested in vivo. So this is how I did.
So I think the person said, was that some sort of software process? So you've actually said this really, that you evaluated some of them in laboratory experimental methods, and some of them were done by looking at the structure of the compound using a software. Yeah. Right. Then the next question, which I can probably answer, but I'd be happy for anyone else to: why are prion proteins resistant to enzymatic destruction with proteinases, and why are they heat resistant, and so on? And as far as I can see, the answer to that question is because of their molecular structure. They have a different molecular structure, and this renders them to have different physical and chemical properties. Unless anyone wants to say anything more specific, we'll probably leave that on one side. The next question was, how do you determine the strain of CJD in a living patient who's been diagnosed with the disease? And I guess that I'd be happy for you to answer that, but my reply, of course, is it depends on what you mean by strain. And as a clinician, I like the biological concept of strain, namely that if someone has an infection, they behave in a certain way. And if they're infected with a different strain of infection, they behave in a different way. I understand the biological sort of behavioral explanation, but it's a bit difficult when you talk about sporadic CJD, which is not in itself an infection. And of course, people talk about, and I think you mentioned this, that the strain behavior is correlated to different chemical structures. So my understanding is that in a living person with CJD in front of you, the only way that you would determine the actual physical structure is because you know that people who behave in that way tend to have this particular structure, or else you would have to take some of the protein from them and in some way study it. But the methods of study are pretty indirect, like the Western blot products or something like that. Have you got anything to add to that or disagree with me about it, or what would you say to the questioner?
Yeah, I agree that the diagnosis of the different strains in human patients, clinical science is definitely one of the first things to look at. But you do also need to look at the prions or PIPC in the brain of the patient, either by Western blot or by looking at the prion deposition in the brain on immunohistochemical staining slides to see where they're deposited, what kind of form they present in a different area of the brain. Those are all kind of things in the look at to determine what type of strain or subtype the patient has. That's what we do normally in our surveillance center on a daily basis. So.
But I think that's the real problem, isn't it? The problem is that you can talk about strain type in behavior. I fully accept that the histological picture, if you look at the brain, the patterns of deposition and the places of deposition of aggregated protein, which of course is, by the time you're looking at it, is PRP res, not really PRP SC, but is correlated with different patterns of behavior. But when you get down to what I think Dr. Ferreira was talking about, and that is that the strain behavior at the molecular level, you're talking about structures of PRP-SC before it's acted on by proteinase K to produce PRP-Res. And the question is that there may be many different species of those as well.
That's definitely one of the problem or complications, because it's new well, many patients actually have a mixture of different PRP-SC types in their brain. Maybe in one region of the brain has one type, in another region of the brain has another type of a different type. So that's the way that we see it pretty often. So that is the heterogeneity of the prion PRP-SC molecules or aggregates in the patient brain is a big problem when you try to see whether this patient has particularly one type or another, or maybe just a mixture. Most likely it's a mixture. Confirmed in a few recent studies.
Yeah. But I think that probably Dr. Ferreira, you have to answer this, but I think that your picture was a kind of way of trying to get this point across in a simple way. You had little different shapes of the sort of abnormal prion molecules, but that's a kind of cartoon representation of trying to get the idea across, isn't it? If you have a living CJD person in front of you, a living person, there's not any easy way that I know of that you can extract material from them and determine exactly what the structure of the relevant PRP-SC molecule is there?
Yeah, right.
I think that was probably the thrust of the question anyway. Was there another? I think there was another question that came out of this, which you can all answer if you like, and that is it the case that research into sporadic CJD will help genetic CJD? Anyone want to answer this?
Well, I believe that the opposite is the right way, so that the study of familial or genetic CJD will help also study in sporadic CJD. But I think that all informations about human prion protein, human prion diseases is relevant to have information and additional evidences for the study also in genetic CJD. So yes, maybe yes, even if I believe that the right way is starting from genetic inherited CJD and then again go to sporadic CJD.
Either of you two want to add to that? Or, I mean, personally,
Sure.
I think all forms of CJD can inform each other. And in fact, the huge interest that's occurred in CJD in recent times is motivated initially rather largely by variant CJD and BSE. It's been a big help to sporadic CJD through that route as well.
I agree with that. Because actually, as you said, they all share some similar features, although they are very different, but they could share quite some common features in the fundamental sense. And for familial genetic CJD, actually can provide probably the best potential to actually develop effective therapy, because it's very hard to find a CJD patient that is at a very early stage. But genetic patients, you can time to some degree when you can give the treatment to see whether something is effective. So in that sense, the familial CJD patient probably give us a window or a chance to actually testing the effectiveness of therapies, which can be used for CJD patients later.
Sure. Two further questions have come through, which I think relate to all of this, and that is that it's interesting now that it's been confirmed that prions can be in the eyes and skin, and maybe there will be a test for strain. And I think, again, it just comes back to, I think, what people mean by strain. And I think that people tend to use the word strain in many different ways here. That's my feeling. But certainly the analysis on available tissues like eyes or skin or other tissues may well help people determine certain things about the protein type and so on. I don't know, anyone want to add to that? But even then, I think it does depend on what you mean by strain. Yeah.
Yes, we are studying the prion in the skin in human patients. That's because the very limited amount of the prion in the skin. I guess we can give some crude kind of inference on what strain it has.
But yeah, you're still
for some subtypes, but not at this point. We don't have a thorough picture of what kind of prion existing in the skin of patients with different SAG subtype. Yeah.
I think that's the trouble, because it does depend on what you mean by prion, I think, in the end, because you're studying aggregated protein that's been processed at that point. That's probably the real.
One of the questions is about the definition of prion strains that you know very well, Richard, that is still very controversial. What exactly defines a strain? So it's kind of evolving concept at this point still.
We won't bamboozle people with that, but I think probably the message is that people do use the word strain in rather different ways. And sometimes people talk about the strain of sporadic CJD versus variant CJD. They're really talking about types of CJD. We've got a very short time left. Does anybody want to suggest how near or how far we are from real treatment?
Well, that's a very tough question. The one that you guys know very well, that there are a lot of difficulties. Laboratory study is one thing. I guess we may have something that may work very well in the lab in the next 5-10 years, maybe. But I think the difficulty of getting enough patients to test the effectiveness in clinical trial may slow down the eventual approval of any therapy. So my guess is being in best hope, maybe in 10 to 20 years, we may have something in the clinic. Best case scenario.
Scientists and doctors always talk about 10 years. I mean, I lived through, I think, 40 years where a cure for MS was always 10 years away. but
is true.
We do need to have hope. And I think what you're really saying is that there are very definite indications of progress here and reasons for optimism, but it's not something that's going to, I think, suddenly change people's lives over the next year or so, is it? It's a bit off. It's
tough. I mean, if you look at it in reality, it's very hard to get a kind of FDA approved clinical therapy or treatment for prions anytime soon. But if you have something very good in laboratory and it's safe, maybe on compassionate treatment basis, probably some people may benefit earlier than 5 to 10 years, maybe even the next few years, probably. But that's just an optimistic hope.
I mean, there were two presentations which centered a bit on genetic side, and I have to say that I'm rather more optimistic about the idea of preventative treatment in genetic mutation carriers at the moment. Do either of Dr. Paleggi, Dr. Ferreira want to comment on that? Do you agree with that or not? That we're more likely to have preventative treatments in genetic mutation carriers than we are to treat people who've got established sporadic CJD.
Okay, maybe they did not catch it.
Can you hear?
I don't know, there seems to be some connection problems. Yeah, Dr. Ferreira seems to be coming in and out, so I'm not sure.
Can you hear me?
It can now.
Yeah, we can hear you, Natalie, but not Anna.
Do you have an answer? Do you agree with that, Natalia? Or?
Sorry, I don't. I lost the last part of the.
It was a speculative question, which I said I personally have much more optimism for a preventative treatment in genetic mutation carriers than I do for a treatment of established sporadic CJD, and I wondered whether you felt the same way.
Yes, I feel the same. I agree with you because treatment, each kind of treatment need to be administered to preclinical in preclinical stage, and so the optimal subjects to treat are the subject at genetic risk of CJD long before the onset of the symptoms. And so I think it is more effective to treat asymptomatic carriers or mutation than sporadic or variant CJD. And I think that when we will find a treatment for asymptomatic subject, this will be also an essential step for the treatment of other human prion diseases. This is my belief, yes.
I believe the sooner we diagnose these patients, more chances we'll have to succeed with the therapy. And also, I believe that the ASOs, the antisense oligonucleotides that have been developed, they are a really good, promising treatment. And by lowering PRPC levels, these could benefit both the genetic cases and also the sporadic case.
Yeah, I certainly agree with you about earlier diagnosis. I mean, that's true for everything, including acute appendicitis, for example. I mean, the earlier you treat something, the better the outcome. I think the worry that I have is that the failure of Alzheimer's treatments, at least in one part, from my perspective, is that we're probably trying to treat people who've got Alzheimer's disease that's clinically evident when they've probably had Alzheimer's disease for 10, 20, 25 years. And so I'm still slightly concerned about how long the disease process has been going on in people who are diagnosed with sporadic CJD. But there we are. Yeah. There's one question that's suddenly come through. Has there been an increase or decrease in people getting sporadic CJD? I think the answer to that question is it's quite a difficult question. There has been an increase over time in the number of people diagnosed as having sporadic CJD. But my own personal view, which may be wrong, but is that that's partly due to an increasing age shift in the population, but actually also much more due to the fact we got much better diagnosis and much better understanding of atypical forms of the disease. And I don't know of any suggestion that sporadic CJD in itself is becoming commoner. Do any of you agree or disagree with that? Or
yeah, I agree with you. It's totally correct what you said.
Yeah.
Okay, we've come to a series of red zeros, which looks very ominous, really. It's having watched Independence Day, I don't know what that means, but still, I think it's at the end of the world. So I don't know, or Chris, are you happy for us to wrap up at this point, or no reply? You can wrap. Okay, that's wrap WRAP rather than wrap RAP, I think. So, well, unless any of you have got any particular other point you really want to make, it's for me to thank you again for your presentations, and I hope that the families who provide your funding see that it's really going to support work that will have outcome, although it's very difficult to predict when that real outcome for treatment will be. So hopefully your work will continue and be successful, and hopefully might even see you next year if we're still alive and the world is normal, which it probably won't be. So thank you very much, and thank you.
Okay, thank you, Richard.
Thank you so much.
Thank you for the excellent introduction and moderation.